and who's the other person? Bolaji, if I'm correct. Bolaji. Genetics. What do you think genetics is? Emmanuel said they are like traits that are passed from organisms to another, or like from parents to their offsprings. So, any contrary opinion to that?
Is the study of genes and hereditary genes and gene variation.
Thank you. So, Emmanuel, that is where you mix it up. When you're talking about transferring or inheritance, that is heredity. Genetics is just the branch of biology that studies inheritance. So, genetics itself is not the transmission of traits. I hope you are with me. So,
Yes,
genetics itself is
yes, ma'am.
not the transmission of traits from parent to their offspring. It studies transmission. So, it's like you're saying something that does something, and something that studies how that thing is being done. So that is the difference between genetics and heredity or inheritance. So while heredity is the transmission of traits from parents, be it in plants or animals, to their offspring, heredity is that branch of science, specifically branch of biology, that studies the transmission of traits. I hope we are clear on that.
Yes, ma'am. We are clear.
Good, good, good. So while we would be discussing genetics, inheritance, and variation, there are some key concepts that we should familiarize ourselves with. So some of those concepts include, the definition of genetics itself, which of course we have established as the study of inheritance, the study of how traits are passed on from their parents to from the parents to their offsprings, and what are the differences that exist among organisms, how it is studied. That is genetics. We've also established inheritance as well as the process by which traits are passed on from parents to their offspring. Now let us look at what variation is. Literally, variation, even without being a biology student or a science student, the literal meaning of variation is when something differs, right? When something is not the same, like there is a difference between one or two instances. So that is variation, differences that exist among organisms of the same species. Despite the fact that scientists have been able to group organisms into different species, this is Homo sapien, this is Panthera, this is Ratus, this is whatever organism that you know, whatever species of organism that you know. Among those species, they earn that species because they have the highest traits, the highest features in common. But then if you observe closely and you look closely, you will still see some what? Some differences that set them apart. This itself is variation. Those differences that occur, that exist among organisms of the same species. That is what variation is all about. As we proceed, probably in the next class, probably next week, we'll discuss more on the types of variation, the different traits that varies in organisms. Okay, then we would not also discuss genetics and inheritance without talking about genes itself. Gene. Those are times we would be talking about, would be talking when we continue with our discussion on genetics. We would always talk about genes. So what are genes? Genes are referred to as the basic unit of inheritance. The basic unit, in fact, they are like what code the traits, what controls the traits that we inherit from our parents. That is genes. And where are they found? They are found within the chromosomes. In fact, they typically make up the chromosomes found in every nucleus of every cell. Genes make up the chromosomes that are found within the nucleus of every cell, and they control inheritance. They control traits that are passed on from parents to their offspring. Then trait itself, we would also be talking about traits. Yeah, the different traits that exist. What are they? There's no how you will talk about genetics. I hope I'm being heard.
Yes, ma'am.
Good. So what are traits? Any behavior, any characters, any feature at all that is likely to be passed on from one organism to another is known as trait or character. Now, while we have several traits in animals, we also have several traits in plants that are inheritable. And what do I mean by inheritable? They can be passed on from the parent organism. Plant, for instance, could possess certain features that when the plant yields seed and the seed is planted, those features that are seen in the parent plant would also be observed in the what? In the offspring. So those are traits. The same thing in man. Height, color, shape and the likes. These are features, traits that can be passed on from parent to organisms. And then we would also be talking about chromosomes. We're talking about chromosomes, those thread-like structures that are found within the nucleus that is made up of the gene, of course. Now, there's something known about chromosomes. Chromosomes are not really visible during ordinary nuclear viewing. When you're looking at the nucleus or you're looking at the cell, you don't normally see the chromosome except when the cell is about to divide. That is when the chromosome become thickened and more visible. So they are known to be thread-like structures that contains or that are made up of gene which controls inheritance. And then how would we talk about genetics without talking about gametes? Gametes, the sex cells, mature sex cells that are involved in the production of new organisms. These sex cells are the ones that actually bring about the fusion of whatever traits that are being combined from both parents. Now, we said gametes are mature sex cells. They are reproductive sex cells. What are reproductive sex cells? Can you give me examples of gamete cells? What are these sex cells that make heredity possible, that make inheritance possible? What are these sex cells?
The egg,
The egg cell. Thank you. And the what?
sperm,
And the sperm cell. The egg cells and the spermatozoa are the gametes. They are the reproductive cells. They themselves have in them traits which are what? Haploid. We will talk about haploid as we proceed. They have traits that are haploid. The cells are haploid cells that they fuse together, and the sperm fertilizes the egg cell to form a zygote which develop into an offspring. And bearing in mind that these gamete cells are haploid cells that bring about, that connect the traits that are gotten from both parent organism. Now let's see some other terms that would also be coming across as we discuss genetics further. Zygote is just the product of fertilization between two gametes. So we've mentioned the two gametes now, the female gamete and the male gamete, the egg cell and the spermatozoa. After fertilization occurs, what is formed is known as zygote. And then the zygote, under favorable condition, when all things being equal, continues to develop into a new organism. We have what we call phenotype and genotype that would also be talking about in genetics as we proceed. When you say phenotype, these are physical observable features that we can see in organisms. And these are not just physical features, they are features that must have been inherited from the parent organism. Height, when two parents of different traits, when they interbreed, and then you see the organism being produced, which of the traits is possessed, is featured, is exhibited by the organism, that you can see is called the phenotype. So any trait that is inherited by an organism and it is visible to our sight, that is phenotype. Observable features, physical features that are seen, that are expressed, emphasis on the word expressed, in organisms. While we have some traits that might have been inherited by an organism, but then they are hidden, they are masked. So both the masked one and the expressed one are actually coded, they are embedded in the gene of that organism. So whatever the organism is made up of in their gene, whatever constitutes the gene of that organism, whether it is expressed or not expressed, is called genotype, the genetic makeup of an organism. And in most cases, the genotype of an organism shows us the phenotype. But then in other cases, we have some genotype. There are some genetic makeup that are hidden, that are masked, that are not normally expressed. So please note the difference between genotype and phenotype. You would always see questions, multiple choice questions about phenotype and genotype. The physical traits that we can see that are expressed in an organism, of course, which must have been inherited, are phenotype, while the genotype are the genetic composition that an organism is made up of. As we proceed, we would see questions where you would be asked to state the phenotypic ratio of a cross between two organisms, and then you would also be asked to state the genotypic ratio of crosses between organisms. So when we get there, we would explore further. Now, what is locus? Yeah, well, we might not be talking about locus more, but it is also a term that is mostly used in genetics. The exact position of the gene where a particular gene is found on the chromosome is called the loci. Loci as the plural form, locus, the singular position. So the exact position of the gene within the chromosome is the locus. Now, as this is said, dominant, literally, dominant traits, the traits that are manifesting, that are superior. Okay, I don't want to use the word, don't let me use the word superior. Traits that are manifesting, that they exert their own influence over another trait. Let's think of two traits now. Let's say color, and then you've crossed between organisms that is white, and another plant with the flower that is yellow. And at the end of the day, the flower produce a seed that has been planted to give rise to another plant with a yellow flower. What happens to the white, the other parent that has the white flower? That means that trait is what? Is hidden, masked. But the one that is dominating, the one that is exerting the greater influence is the yellow flower. So that yellow trait is what? The dominant trait or the dominant character. And in most cases, dominant characters are always controlled by dominant gene, but except in the cases of partial dominance, which we would also be talking about. Dominant characters are characters or traits that exert influence, or that are seen, that are expressed in the presence of what? Another trait. Two traits now are combined, and then one is showing while the other is not even showing at all. So the one that is expressed, that is manifesting, is the dominant character. And please note, it is always controlled by the dominant gene. We've established what dominant characters are. How about recessive characters? So it is just the opposite of a dominant character. We have two characters that are being considered, that are being crossed, and one is dominating. So what happens to the other one? Is hidden, is being masked, is being covered. That is a recessive character. A character that is what? That is hidden, that is not seen, due to the presence of what? A dominant character. And just like the dominant character, recessive characters are also controlled by recessive gene. We would continue with other terms. We would be using this word often as we proceed. Homozygous, homozygous, heterozygous, homozygous. In genetics, traits that are seen in organism are combination of traits from both parents. We mentioned the gamete cells. Each chromosome in every cell has or code for a particular trait. The number of chromosomes in organism differ depending on the organism we're talking about. Let's take for instance man now. The number of chromosome in man is 46, which is what? 23 pairs. Chromosomes are always in pairs. 23 pairs. Now these 23 pairs control several traits, several characters. One of these character is the sex itself. The sex of a child, either a child would be a boy or a girl, is controlled by one pair of these 23 pairs of chromosome, and those are the X and Y chromosome. Now in every gamete cell, the sperm cell and the egg cell, there is always a half pair of chromosome. You know, we said we have 46 pairs of chromosome in a complete organism, right? In a diploid cell. But the gamete cells have half half number of chromosome called the haploid cells. These chromosomes are now always being arranged in pair. Whether a cell is carrying the X chromosome or the cell is carrying the Y chromosome, they are always arranged in twos. Now, if the gene that is portraying or that is representing a particular trait is the same gene, like we have X and X now, that is what? Homozygous, whatever thing, that organism, probably homozygous, tall, homozygous, whatever trait we are considering. If that gene is not the same, like probably you have X and then you have Y. I'm just using X and Y because that is what I've cited as one chromosome first. In most cases, traits are mostly represented by the first letter of the trait we are looking. If you are considering tallness, so they would use the first letter for tallness, which is T, the uppercase, representing the dominant trait, while they would use the lowercase representing the recessive trait. That is the contrasting trait of tallness. What would be the contrasting trait for tallness now?
Short.
Shortness. So if you had used the capital T to represent tallness, then the one that is recessive, that is being marked, would always be represented by what? Small t. And I've mentioned earlier that these chromosomes are always in pair. They are always in twos. Now, if an organism is said to be tall, it is either that organism is possessing within it a genotype that is what? Capital T and capital T. Sorry, my screen is acting up. Okay, capital T, capital T. In this case, that this organism is exhibiting this tallness trait, if you look at the T, is the same. The same gene for tallness is what make up that tallness trait in that organism. Here it is said to be what? Homozygous. Because the same gene is what? Is controlling that trait of tallness in that organism. But in a situation whereby an organism has inherited one haploid T cell from the father, and then the organism also inherited another haploid smaller lowercase T cell from the mother, at the end of the day the organism would still show the tallness trait phenotypically, even though the gene is different from this type of gene, this type of tallness that is homozygous. So when we have two types of genes that are not of the same type representing or coding for a particular trait, then we say it is what? Heterozygous. So when you have two similar genes controlling a character, homozygous. When you have different genes controlling a particular character, we say it is what? Heterozygous. Then you say that trait is heterozygous. So as we proceed, we'll be talking about, okay, homozygous tall, heterozygous tall, homozygous short. So you should not be confused. In fact, in most questions, they would ask you, they will tell you that homozygous, two heterozygous parents. So you should be able to understand and interpret the term heterozygous to represent your gene whenever you're crossing. Can we proceed? Now we move on to allele. They ask questions a lot about the definition of alleles in multiple choice questions. Allelomorphs are also called allele. They are pairs of genes that code for a particular character. They control a particular trait. So when you hear alternative form of genes, in fact, think no further. Just think of allele. Some people call it allele, some people call it allele, whichever way you've chosen to call it. Just make sure your spelling is correct. That is another thing I need to mention. In biology, spelling must be correct for you to score, for you to have your mark. If you know it and then you've done one substitution somewhere or omission or addition anywhere in your spelling, my dear brothers and sisters, you're wrong. You would not be acknowledged. And then you'll be like, actually, I got the answer correct. Now it's just one letter, one sound that I did not have or that I added. You are not correct. So please note your spellings as you study biology. Note your spellings. Allele is spelled Allele, or you can spell it in full, allelomorphs. So the alternative form of gene that control a particular character. Now, what are hybrid? This one is even commonly used by non-sciences. Hybrid. I want hybrid. So whenever you hear the word hybrid, it's just the product of a cross between two parents of different character. You have two traits that you're considering, and then you want a combination of both traits. You want to see you have an organism that has good, probably that give good yield, and then you have another one that also have another better yield, and then you are trying to what, put them together so that you have what, a better result. Let's say for instance you have a plant that produce a fruit that is very big. Wow, that is a plus. We would always want a plant that produce fruit that is big. But the plus there is that even though the fruit is big, the fruit is not sweet. It is sour somehow. And then you have another plant that produces fruit that is small. But you see, as small as that fruit is, oh, it is very yummy, sweet. So what do you think, as someone that has learned genetics, that you know that you can actually crossbreed? So you cross between a plant with the big and the big but sour fruit, and then the small but sweet fruit. At the end of the day, you would have one offspring that would inherit the big traits and the sweet trait from both parents, and that organism would now manifest the big and sweet taste. That organism that have combined the desired trait in both organism is a hybrid, whether the trait is actually desired or not. So far it has combined trait from both parents, that organism is what, a hybrid. I hope we are together. So it is the result of a cross between parents of different what, of different traits, of or different character, or of different genotype. Combining the genotype from both parents now is giving us hybrid. Now as we go on with hybrid, we'll be looking at monohybridization or dihybridization. Sorry, before we continue, we have hybridization here. You are all English students, of course. You are all in grade 11, 12, that's SS3. So you know, you should be able to differentiate between hybrid and hybridization. Hybrid itself is the product, is the offspring, while hybridization is what, the act, the process of what, creating a hybrid. I hope that is clarified enough.
Yes, you are, ma.
Good. Hybrid is the product, is the offspring, is the result of the hybridization, while hybridization is the process of what? Of crossing two parents of different traits, desired traits. Now we can have hybridization in twos. We can have monohybridization. That is if you're control you're concerned about a particular trait, one contrasting trait. Probably you are looking at color, white and black, green and yellow, or whatever trait you are looking at, just one contrasting trait. That is monohybridization. You are only considering one trait. But when you talk about dihybridization, you are control you're considering more than a trait, more than one contrasting trait. You are looking at the height of the plant. At the same time, you are putting in consideration the taste of the fruit. So that is two traits. Either it is sweet or sour, then the other trait, either it is tall or short plant. Or you are looking at the height and the color of the flower. Or you are looking at the color of the flower and the shape of the seed. You know, it is either the shape is round or the shape is wrinkled. So that is one contrasting trait. And at the same time, you're also looking at, okay, should the flower be yellow or green? So that is dihybridization. When you're considering hybridization between parents of two contrasting characters. I hope that is sorted and clarified. Monohybridization, dihybridization. When we talk of the law of genetics, we would be talking about mono and dihybridization too. The first law of genetics is mainly concerned with monohybridization, while the second law of genetics is mostly concerned with dihybridization. Now we have another term here, mutation. Mutation. Yes, when we talk about genetics and variation, we would always talk about mutation. Yeah, there is like a blueprint now, that okay, when you cross organism of different traits, expectedly one of the traits will dominate. That is what we have been saying since abi. One would dominate, the other would be masked, will be hidden. Or when you cross between two organisms of this and this traits, of course you are crossing two traits, you know that the offspring, the hybrid the header of the trait that you are considering. But what about in cases whereby you have crossed a tall and a tall plant? You have vetted that the two tall plants do not even have any traces of shortness. And then at the end of the day, you have an offspring that is short. So if it is that the two plants they're having a trait of shortness, we would have think, okay, probably the shortness trait now became, the two recessive traits now became dominant. No. In case of mutation, you just have a kind of a deviation, a aberration from the norms, a sudden change in the gene of an individual or in the gene of an organism, resulting to what? A totally different feature or character that is different from what? The parent organism. And then you would wonder, how come this organism is like this? We do not have any of these traits in the history, in the lineage of this organism. So that is what mutation is all about. A sudden change in the form and structure of a particular gene that gives rise to what? A new trait. Now, there's something we should also note about this mutation. Mutation is not just something that happens overnight. It is not that, okay, oh, my mom and my dad are dark. None of our family is dark. And then the following morning, I just give birth to a child that is what? That is fair. It is not. It must have been something that is what? That has been accumulating for a while, and then it would manifest after what? A whole lot of years, or I don't know. Is there another time to describe years? Millions of years. So sudden change in the form and structure of a genetic of a gene that give rise to what? A new character that is entirely different from the existing character. That is mutation. And then please note, mutation is one of the factor that leads to variation. That differences that exist among organisms of the same species. We have the same father now. Myself and my sister and my siblings, we have the same father, but then we have some things that differ, probably in terms of our hair structure, the shape of the ears or whatever. We shall have that thing that differentiates us as siblings. So it is one of the things that leads to variation, that mutation that we've talked about. Okay, let's see, let's continue. We've talked about most of the terminologies that would be using, that will be coming across: dominant gene, dominant character, homozygous, heterozygous, blah blah blah. Now let me take this slide away. Let us see those traits that are, or that would be looking at, that are inherited in organism, those traits that can be transmitted from parent to offspring. So this is how we're going to do this part. We are four presently: Mariam, Emmanuel. I should mention Emmanuel for, because he joined the class first. Emmanuel, then Abolaji. I'm not so sure about that name. Abolaji, is that it? The second person that joined, Ola something. And then we have Olamikun Okpe and Mariam. So that is the order. So you are going to give me one trait each that can be passed on, or character that can be passed on from an animal to its offspring. There are another one trait that can be passed on from a parent plant to the offspring plant. Just one transmittable trait in plant and animal. So let's start with Emmanuel.
One transmittable trait. I think in plants, I would say color.
Color is brilliant. You're correct. Color, that is in plant. Okay, one transmittable trait in
Then
animal.
in animals, I would say height.
Height. Okay. Now let me go back to that color, so that we would not limit those that would be responding. Color is actually it is encompassing. When you say color, what color? Is it color of the leaf? Is it color of the flower?
Okay,
Is
color of flower.
it
Yes,
color of the
color of
seed? It could
flower.
be color of the hair. So color of the flower. Thank you, Emmanuel. So let's go to sorry, let me look for that name. The person is not correcting me. I've been trying to call the person's name and he or she is not correcting me. Okay, it's Bolaji. It's Bolaji. Bolaji, yeah. Mention one transmittable trait in animal and one transmittable trait in plants. One transmittable trait in animal. I hope the person is still with us. Okay, I'm
One
listening.
transmittable, Untransmittable traits
Now
in
she's
animal
laughing. Okay.
is just like their skin, the color of their skin.
Skin color, that is correct. Emmanuel mentioned height. You mentioned skin color. That's correct. Okay, what about what other trait do you think can be transmitted in plants? Aside color of the flower that Emmanuel has mentioned.
Like you said, the sweetness in fruits and
The taste, that's the taste. Food,
yes,
taste
ma'am.
of the food. Taste of the food, you're correct. Okay, let's have you, Olamikun. One trait in animal that
Trait in
Olam
animal eye color.
eye
Trait in
color.
animal eye color.
Okay. In
Trait
plants.
in plants leaf pattern.
Patterns of the leaf. That is brilliant. Okay. Mariam, let's have you. One trait in animal. You should there? And one trait in plants that is transmittable.
And blood type in animals and height or shape in plants.
Height or shape of the plant. Blood
Yes.
type in animal. That is very, very correct. Thank you, everyone. Well done. Well done. Well done. Now let's see what we have here. Traits that are transmittable in animals. We have body posture or shape. We have shape of the head. Should not surprise you. If you look at some people, you see the exact forehead of either their mother or their father, you'll be like, kilo de. Kilo de, is your daddy running away? The shape of the head, the shape of the nose, the shape of the ears, shape of size of the nose, head, ears, color of the skin, color of the hair, color of the eye, the voice. When some people talk, you would think it is their dad or their mom. That I have a sibling, my immediate sister. When she talks, you would think it is my mommy that is talking. You would hardly differentiate. So these are traits that can be inherited. Voice itself. Then the intelligence, yes, intelligence. I don't want to be stereotypic here, but there is this popular. I don't know if it is actually a fact or a myth that children most of the time inherit their intelligence from their mom. Please do not be offended, but I choose to believe that children most of the time inherit their intelligence from the mom. So intelligence is also a trait that can be passed on from their parent to their offspring. Height, of course, we've mentioned that earlier. Blood group, like someone rightly mentioned. Baldness, yeah, this is also inherited. In fact, it is sex linked. We'll talk about some sex linked traits. So when we say sex linked, these ones are only controlled by the X chromosomes in man. We've talked about the X and Y chromosomes earlier. Those chromosomes that control the sex of a child. So some of those chromosomes carry another trait in them that the only time you get to have those traits is when you inherit it from that sex chromosome. Baldness is one of those. Hemophilia, sickle cell and the likes. Color blindness too. So baldness, tongue rolling, ability to fold and roll the tongue. It is a trait that we inherit. So if you cannot roll your tongue, sorry, it's because you did not inherit it. But if you can roll your tongue, definitely either your mom or your dad must have been able to roll their tongue. It is also a trait that differs in organism. We'll talk about it when we talk about variation too. Sickle cell anemia, the sickle shell of the sickle shape of the red blood cell, such that it won't be able to transport enough oxygen, and that is why you see individuals that are sickle celled, they have crises with they used to have crisis because there is not enough transportation of oxygen because of the way their red blood cell is what sickled. No enough space to carry oxygen and transport it within the body for energy production. Hemophilia, yeah, inability of an individual's blood to clot after the sustained injury. These are traits that can be inherited. Color blindness, inability to distinguish between colors, of course, obviously. We have some cells that controls the vision, the white and color vision in the retina, the rod and the cone cells. So you see these cone cells, some people do not have it, they do not have enough cone cells inherited already. So cone cells are responsible for color vision, for you to be able to distinguish between colors. Yeah, fingerprints, of course, that is actually a unique character, but then we still have whatever fingerprint that is manifested in us is a combination of prints that we must have inherited from our parents. Ability to taste PTC. Who knows what PTC means here? PTC. He said that ability to taste PTC is also an inherited trait, transmittable trait. What is PTC? You must have heard PTC. We'll talk about it in variation too. It is a discontinuous variation. It is either you can taste PTC or you cannot. If you can taste it, you find it to be bitter. And if you cannot taste it, it is just tasteless, just like that. So that means you cannot taste PTC. You do not inherit the ability to taste it. So what is the full meaning of PTC? Who knows that? Should I give you as an assignment or I should just tell you now? No, we'll be meeting next week. PTC is a chemical known as. Okay, someone wants to talk. PTC is phenyldiocarbamide. It is a chemical that some people find tasteless, while some people find it bitter. So that ability to either taste it or not taste it is also inherited. So if you cannot taste it, that means that is gotojogun, that is what you inherit. And if you can taste it, well, probably either your father or your mother or one of your family, your ancestor can taste it. So these are traits that are transmittable in animal. And then we have some traits that are transmittable in plants as well. Height of the plant, like we've mentioned. Nobody mentioned the size of the fruits. Size of the leaf, I think it was mentioned. Taste, someone mentioned taste too. The content of the food content of the food. The color, I think we've mentioned color of the leaf, flower, fruit, and seeds. Resistance, how susceptible is the plant to diseases? How resisted, how do you speak English? How is the plant being able to resist diseases? So if a plant is vulnerable to diseases, then you say the plant is what? Vulnerable, abi? It is, the plant would easily contact any disease or any pest or whatever. And that ability can also be passed on to the offspring. But if a plant is able to resist disease, that is a good trait that farmer would what? They would want to ensure that it is passed on to the next plant. So that itself is also a transmittable trait. Shape of the leaf, fruit and flower, then the lifespan, the growth habits of the plant. These are some of the traits that can actually be what? Passed on from parent to their offspring. Now let us look at the work of Mendel in genetics. So this is where the main work is now. We're doing a whole lot of cursing and cursing and cursing, and then we'll attempt questions here today, God's willing. Now, this genetics that we've been shouting, and this dominant trait, and this homozygous, that heterozygous. Why genetics? What led us to genetics? Why are we disturbing ourselves? Well, it is because scientists, as they are, when you did science in GS1, you were told that science is a systematic means of what? Looking for solutions to problem, a systematic approach towards providing solutions. Why do you need solutions? Because you did not sit in a place, right? You're trying to explore, you're making inquiry, you're finding. So in a way to also provide solution, this man, Gregor Mendel, he observed that traits are actually passed on from parent to offspring. And then in a way to provide better answers, he now studied, he experimented with a particular type of plant, the Pisum sativum. Please note, when you see this in question, they might not say pea plant, they will just use the scientific name, the Pisum sativum. So you should know that the Pisum sativum is also the garden pea or the pea plant. So he has observed that traits are passed on from parents to offspring. That is inheritance. So he now studied that inheritance, that transmission of traits from parents to offspring. And what plants did he choose? He chose the Pisum sativum plant. And because he was the first scientist whose work was documented, or that was established, that okay, he was the first person to study and research on transmission of traits, then he became what? The father of genetics. Please note that too. They ask a lot. Who among the following scientists is referred to as the father of genetics? Gregor Mendel. In fact, the first and the second laws in genetics are credited to him. Mendelian first law of inheritance, Mendelian second law of inheritance. Those laws also have another names that would also talk about when we get there. So where are we? He experimented on Pisum sativum, and then he observed how traits are passed on from parent plant to the offspring of the Pisum sativum, and then he chose this plant for a reason. They used to ask questions about that too, that what is the plant used by Gregor in his attempt to experiment transmission of traits. Pisum plant, right? Pisum sativum, or the pea plant, or the garden pea, whichever one you chose to remember it as. But I would advise you should remember it as everything, because either of those name could be used. Now, why did he use the Pisum plant? They used to ask that too. Why? He actually used this plant because of three reasons. I don't know if any of you had come across one of the reasons he used Pisum plants. You know, when you want to do something, you try to go for what is convenient. Okay, what will make this thing easier for me? What will make it easier for me? And that is why he considered Pisum too. He chose it, one, because this plant is an annual plant, a plant with a short lifespan. That I will not spend the whole year trying to study I will not spend millions of years trying to study the plant, waiting for the plant to produce seed, and then the planting the seed and seeing the result. You know, if you are choosing a perennial plant, that might take years for the offspring to be seen, right? Another reason he chose the Pisum plant is because the Pisum plant is also a plant that has many contrasting characters, traits that are alternating: height, like tall and short; color, like red and this; shape of the seed, like round and wrinkled; taste of the fruit, like bitter and sweet. Different traits that you can even consider. So it is a plant with what? Many contrasting traits. I can, okay, if I don't want to study the height, I can actually study the shape of the leaf. Okay, I can actually look at the color of the flower. Okay, I can actually look at the taste of the pea seed itself. So he chose it because it is a plant with many contrasting character. That is the second reason. Then the third reason he chose Pisum plant is because it is a plant that is self-pollinating. Self-pollinating. You've heard of the word pollination before, right? Pollination. You must have done pollination
Yes,
in
ma'am.
SS2. Pollination. When you learn plant reproduction, you would kọ́kọ́ learn. Sorry, pardon that my choice of English. You will kọ́kọ́ learn pollination. So pollination is when pollen grain, the male gametes now in plants. You know, you mentioned the male gamete in animal, the spermatozoa and the egg cell. So the male gamete in plants, so the pollen grains, the way when they are transferred from the anther where they are found, or where they are stored, to the stigma of another plant, or of the same plant, as the case may be, to go and pollinate the ovule, which is also the female gamete in plants. So that is pollination, transfer of pollen grain from the anther to the stigma. Now, in some plants can self-pollinate themselves. Like the anther of that plant, one flower of that plant would pollinate the stigma of the same flower, or of another flower but of the same plant. If it is just one plant pollinating itself, that is self-pollination. But for cross pollination, you would need two plants: the anther of a flower from a plant pollinating the stigma of another plant, not of the same plant now. So for self pollination, it can be the anther, the pollen grain of the anther from a flower on that plant pollinating the same flower. Like, let's say this is the plant, this is one flower, this is another flower, and then this is another flower. So in a situation whereby you have the pollen grain on the anther here pollinating the stigma on the same flower, it is self pollination. Or the pollen grain of this flower pollinating the stigma of the second flower next to it, in as much as it is still of the same plant, it's what? Self pollination. But if the pollen grain moves to another plant, that is cross pollination. So that might be difficult. So he now chose the pea plant because it is a self pollinating plant. The flower can pollinate itself, whether any of the flower on the same plant. So those are the three reasons he used the pison plant. Please do not forget that. They used to ask too. Which plant is used by Gregor in his experiment on genetics, or in his experiment under of inheritance? Why did he use the plants? Those are the three reasons. It is an annual plant, it is a self-pollinating plant, and it is a plant with what? Several contrasting characters. Any question before we move on? Because we are going to be looking at caution, and before we look at the laws. Any question God did with the pison plant? For him to experiment, he chose a pea plant that is tall, and then he crossed the plant, like he planted the plant for several years to ascertain that that plant is indeed a tall plant. Do you know why he did that? In sciences, we always control our experiments. When you are reporting an experiment, they tell you that what are what are the variables that were controlled so that your experiment will be accurate. So in his own bid to also ensure that the experiment is accurate, he did not want a situation whereby he just picked a plant that is tall, but not unknowingly, okay, that plant might be a tall plant with what? With a hidden shortness trait. So he does not want story that touches like that. So he just what? He planted the same plant continuously to ensure that, okay, this plant is indeed a tall plant. So definitely that plant is what? A pure tall plant breed. A pure tall plant. We've established why we use capital T to represent tallness, right? And I also mentioned that genes are always in two, allele. They're always in two. They're always in two. When they are the gametes, they are always in two. The genes that are found in the gamete are always in twos. So he established that that plant is indeed a tall plant. Then he got another short plant, and then he planted it for several years again to ascertain, to be sure of the fact that that plant is actually what? A short plant. So what are we using to represent our short plants now? The lowercase letter for what? The T. So now there's something you should note. Anywhere you see an uppercase letter representing a particular character or gene, that uppercase is always the dominant gene. That is always dominant. And whenever you see the short or lowercase letter, it is always the what? The recessive gene. The one that would be masked, that would be hidden. I hope we are together. I need response. So you didn't answer my last question where I say should we proceed or not. Are we together? Good. Now after he has ascertained that
Yes,
the
ma'am.
plant is indeed a tall plant and the other plant too is indeed a short plant, he now what? He pollinated the tall plant with the pollen grains of the short plant. He did the same with the short plant too, pollinating the short plant with the pollen grain of the tall plant. And now what did he have? Let's see what he got. Now if you are doing this in genetics, let me reference this. This last exam that the result just came out on Monday. During the course of marking their exam coordination, there are some rules that were emphasized, and it would actually take a toll on most learners. Now what is that rule? Is that when you want to show a cross in any inheritance experiment, you should always indicate your parents. You should always label. So the first gamete here, this is the gamete, the gene representing the what? Oh sorry, the gamete that contains the gene representing the tallness trait. Well, this is the gamete of the second parent that is representing the recessive gene for short trait or for shortness trait. English people. So you should always label your gamete. There is a mark for that. This is what? Your parent's gamete. Or you can just put parents or whatever, parent genotype or parent genotype. This is the parent genotype now. Then after that, you show that there is a what? Cross. You are trying to fertilize or pollinate this with this or this with this. So that cross sign is very important. You know the implication of candidate for this year WAEC that did not put this cross sign. gbogbo 8 marks yẹn gone. It is mentioned that if this cross sign is not there, their working should not be acknowledged. Imagine going through that mile, and just because you did not cross, you did not show that you are crossing the parent's genotype, you are wrong for everything you have done. So please always note. Now, immediately after you've shown your parent's genotype, the parent organism that you're crossing, you also bring out their gametes. Gamete T, then the other gamete C. You don't need to put cross here again. Then for the next gamete T and the next gamete C. Now here too, according to WAEC, you have 2 options. It's either to leave it apart or you circled it. So if you are not circling, you make sure that the 2 gametes are what? Far apart from each other. This small t and this small t should be more far apart. So I would advise, because how would you define far apart? You might think you have put it to be far apart, and they would say, okay, it's not far apart enough. So it's safer to what? To just circle your gametes. So you've identified your parent genotype, you've put your gametes, make sure you circled your what? Your gametes separately. There's a term that we did not describe when we were talking about terminologies. We'll talk about that term now. Don't know how that skipped our mind. It was in my previous slide. Okay, don't worry, let's continue. So now when you cross these 2 gametes, this T would multiply this T. So the offspring that would be gotten would be an offspring that is what? That carries the capital T, small t. I don't know if you are with me. Then the T again would multiply the second gamete of the second parent, and then we would have what? Capital T. This is capital T, then small t. Then you come to the second gamete of the first parent, the capital T, to multiply the what? The first gamete of the second parent. That is what? Capital T, small t. And then the second gamete again with the second gamete of the parent, of the second parent. That is what? Capital T, small t. This is a simple case of what is it called in expansion in mathematics. Just multiply across. Your first t here multiplied by the 2t, then the second t by the 2t, then you have your offspring. Now these are the first offspring. Now these offspring are called F1 offspring. This is what I said we omitted. F1. This one too is very important that you label it to have your mark. To have your mark, your complete mark. F1. So these first offspring are called the first filial generation. The first filial generation. Now what did Gregor observe? What did he observe? After he has pollinated the flower of the tall plant to the short plant, he noticed that when he planted the seeds, all the offsprings of the F1 are what? Are this type of tall. They are tall, oh. This thing you are seeing here, this capital T small t, capital T small t, capital T small t, capital T small t, they are all tall plants. Now the thing is, he did not know what their genotype is. He only saw that shebi tall plants and short plants pollinate. How am I having? I hope there is no one here that does not understand Yoruba. Is there anyone that doesn't understand? Do you understand Yoruba? No. Pardon me for that. Oh, I didn't even hear your response. Now, have actually crossed between a tall and a short plant. How are the offspring now tall? Not knowing to him that this type of tall offspring are heterozygous tall. They are not the same type of tall as one of the parents here, which is what? Homozygous tall. This one is homozygous tall, the parent's genotype here. The other second plant too is what? Homozygous short. But at the end of the day, he got 4 plants, offspring that are what? Tall. But the type of tall that they are, he does not know. He shall know that they are what? They are tall. Meanwhile, they are all heterozygous tall. So while he was now concerned that, okay, what happens with the F1 generation? Why is it that they are all just like the first parent, all tall? So in order to establish the type of tall that they are, these offspring are, he now went ahead again to cross between offspring of what? The first filial generation, the first offspring. So that time that I said we did not discuss, that is the filial generation. When you hear the word filial generation, They are the direct offspring of a what? Of a cross between two parents. So the hybrid, the first hybrid are always the F1 generation. They call them F1 or first filial generation. Then the offspring of those ones now, grandchildren, abi, they are the F2 generation. The great grandchildren would not be what? No response.
F3 generation.
Thank you, F3. But in most cases, we actually don't usually go to the essence of looking for the F3. But I've seen questions in IGCSE exams where candidates have been asked to show the F3 generation. So that would be the offspring of the second filial generation. So he now went ahead to cross the offspring of the first filial generation. And what would he get? Now let's get this erased and then see what the offspring. Don't not your F1 generation. Oh. Heterozygous tall abi, capital T small t. Those are the offspring that he got for the F1 generation. Why is my screen not erasing very well now? Okay, we are good with that. So for the F2 generation now, the parent organism, the parent genotype would now be what? Capital T small t, capital T small t. I would give you assignments to be submitted before the next class. Under crossing, capital T small t. Then this one would now be what? Your P, or you can write it in full. Parent gamete. Why is this one skipping? Don't forget your what? Your cross sign to show that you are crossing between the two parents. Your cross sign. This one does not want to write in full. Then the gamete now. Your uppercase G. This allele for tallness. Then the other one. Then this one. Then this one. Now the F2 offspring now, the F2 second filial generation would now be this and this. What type of offspring would we have? Homozygous tall. Homozygous tall. The same gene is representing this trait for tallness. Then the second one. What type of offspring? Heterozygous tall, ba? Still tall, oh, but not the type of tall that the first offspring is. Then the second offspring here, capital T. Always write the dominant gene first. That gene that would manifest, always write it first. Capital T, small t, heterozygous tall. Then the last one. Yeah, sorry, my crossing is not beautiful. It's not neat. Then we have a cross here that is what? Short. Now we now have a short offspring. The first time he did it, the first offspring, none of the offspring were short. They were all tall, despite the fact that one of the parents is what? Short. The only time he got a short offspring is what? The second. It now manifests that recessive trait that was hidden. This small t that was hidden here from the first generation, it now became manifested in what? In the second generation. We now have one short plant, one short pea plant now. So at the second filial generation, he got 4 plants with 3 of them being tall and one that is what? That is short. Don't forget the first time all were short, but what type of short were they? Homozygous short. All were tall, homozygous tall. So they could ask you, what is the genotype of the first filial generation of Darcos? They were all heterozygous tall. The offspring, they have different gene, capital T and small t. They have capital T and small t for the first filial generation. So they were all heterozygous tall. And what is their phenotype? What were we able to see? The offspring were all tall. So the phenotype is what? Tallness. No short plants. No short pea plant. You didn't see any short pea plants for the first filial generation. So for the first offspring, the one we did earlier, the phenotype was all tall, 4 tall plants. And the genotype were what? 4 heterozygous tall plants. But looking at this F2 now, how many tall plants do we have here? I want you to respond. How many tall plants did you get?
3.
3. This one is tall.
3.
This one is also tall. This one is also tall. So he got three tall plants. So the phenotype of the second filial generation would not be what? Three tall plants and what? How many shots? Just one. One short plant.
One.
So the phenotypic ratio would not be 3 to 1. Out of 4 offspring, 3 are tall, 1 is short. So that is the phenotype that we can see. What we can I think we shall can see that 3 are tall, 1 is short. Now, despite that 3 of them are tall, they do not have the same gene, the same genotype. Their genetic makeup is not the same. So what is the genotype of this one? Who can respond? What is the genotype of this one? How would you describe the type of tallness in this first offspring here?
Um, One
Homozygous.
homozygous
Thank you.
tall,
Thank you. That is
two
homozygous.
heterozygous.
So that means we have one homozygous tall. Now, this one is what? What type of tall is this one? Heterozygous, right? Because we have different genes that are representing that
Two
tallness.
heterozygous tall.
Yes, two are heterozygous tall. So that means one homozygous tall, two heterozygous tall, and then we have what? One homozygous short. One homozygous short. So and for the second offspring, he got the phenotype of 3 tall and 1 short. But the genotype he got was what? 1 to 2 to 1. One homozygous tall, 2 heterozygous tall, and 1 homozygous short. So that is what he got with the second filial generation. That now led him to the theory, to that law that so this trait for shortness that was not manifested in the first filial generation, it was still there, it was hidden, and then it became manifested in the second filial generation. That means the gene for it was only hiding. It is not that it become altered. No, it was not altered. It was only hidden, and then it became manifested after it has seen about another recessive trait rather for it to become manifested. So that was what led to what is first law of inheritance. That law is now termed the law of segregation of gene. So they can call that law. We have there are 3 names that you could refer that could be referred to, or that law could be referred to. You could be asked in 3 ways. They could ask you to state the first law of Mendelian first law of inheritance, or they could ask you to state the law of segregation of gene, or they could ask you to state the law of monohybrid inheritance. Why is it monohybrid? Because they only considered the height here, just one contrasting trait, tall and short. So it could be said to be what? Monohybrid law, or the law of monohybrid inheritance, or the law of segregation of genes, or Mendelian first law. And what does the law say? The law says that genes are responsible for the development of individual character. Whatever character that you have, that you must have possessed, your gene is responsible. Whether it is homozygous or heterozygous, your gene is what controls your character. So he said genes are responsible for the development of what? Individual character, and they are independently transmitted from one generation to another without undergoing
Ma, please,
any alteration. Yes, I'm
sorry, ma.
listening.
Can you come? Okay, I've seen the law. Like, I wanted
Genes
you to
are
come again with the law,
responsible
but
for
I've
the
seen it.
development of individual character, and they are independently transmitted from one generation to another without any alteration. Ó lè pẹ́, ó lè yá, that gene that is within it will still what? It will still manifest itself. That is the ideal, that is the basis. Well, except in the cases of mutation, which we would be looking at when we talk about variation. So that was what led to what? Gregor first law of inheritance. After he has crushed the plants, the first filial generation all produce tall plants. Him not knowing that they are not the same pure tallness as that of the parent tall. So when he now crossed the offspring of the first filial generation, he now got a what? The shortness trait that was a trait of one of the parents. So then he now realized that so the gene never get altered. They were what? They were only not manifesting in the first offspring, and that led to how he established his first law. Genes are responsible for the development of individual character, and they are independently transmitted from one generation to another without any alteration, without undergoing alteration. Now we are supposed to look at the second law of inheritance. And I really
Sorry, I can
like
ask a question.
yeah, I'm listening. I'm listening, I'm with
Sorry,
you.
what's the other name for the law of the segregation of genes?
First law of Mendelian first law or monohybrid law. Mendelian first law or monohybrid law. Or they could say first
Okay,
law of
ma.
genetics or Mendelian first law. When you hear monohybrid inheritance law or law of monohybrid inheritance, you see the same thing as law of segregation of gene. The second law is the law of independent assortment or dihybrid inheritance, or the law of dihybrid inheritance, or Mendelian second law, or second law of inheritance, whichever. Why is it just second? Or dihybrid, or what? That is independent assortment. Well, I would want us to attempt questions, more questions on that first law before we move ahead. If you do second law of inheritance. Now, what happened with Mendel's and how did he come about the law of independent assortment? Then he now went ahead with plants with two contrasting characters. He used a pea plant, so still a pea plant, that is the seed, sorry, the flower is yellow, a yellow flower, and another one with a green flower. So what would be the gene for green now when we have capital Y for yellow? What would be the gene for green now?
Small letter Y.
Thank you. So small letter Y. Now, some people might get confused that green, that should be small letter G now. Well, you will be confusing yourself more if you use small letter G. So it's better to stick with one letter. If you have chosen one letter to represent your gene for a particular character, go ahead with the lowercase of that same letter for the what? The contrasting character. So the yellow pea plant was crossed with the green pea plant, but then the type of yellow pea plant that he used was the same yellow plant that has a what? A round seed. So the round was dominant over the other seed, which is what? Wrinkled. So he used a yellow plant, a yellow pea plant, sorry, a pea plant with a yellow flower and a what? That will produce a round seed. And then he went ahead with what? The green flower that usually produce what? A wrinkled seed. Don't forget, always in pairs. Always in pairs. So if we had used uppercase R to represent round, then wrinkled, instead of representing it with lowercase w as wrinkled will start with its first letter, we would use it with the lowercase of what? R. So here is wrinkled. Now what did he get? For all the first offspring that he got, after he crushed this plant that is with a yellow and round yellow flower with round seed and green flower with wrinkled seed, all the first filial plant that he got were all yellow and round. Please note this is the hybrid because we are looking at two traits. So all the F1 plants were all yellow and round. But please, this is not the type of yellow and round that they would be, because they are being crossed today. They will be what? Heterozygous. Capital Y, small y, capital R, small r. This is the type of plant he got for all the first filial generation of the cross that he did with the yellow and round and the green and wrinkled pea plants. Now when he now did that, he now observed that ideally, just like the first monohybrid inheritance that he did, the gene that are responsible for those traits should still be what? Should still segregate. They should still segregate. So he now expectedly proposed that, okay, the offspring that he should get, if those genes segregate, The offspring that I should get would be an offspring that is what? Yellow and round. Yellow and round. Sorry o, my screen is acting up. Yellow and round. Just take it in your mind that is round. Then you should also get a plant that is yellow and wrinkled. I think it's this part of my gadget that is not want to be writing. Wrinkled. Wrinkled though with ELD. Then you should also get an offspring that is what? That is with a green and round. Green and round. And then you should also get what? One that is what? Green and wrinkled. I hope we are together. So he proposed that the segregation of those genes should provide offspring that would cover all the traits. One that would be yellow and round, one that would be yellow and wrinkled, one that would be what? Green and round and green and wrinkled. And what did he do to actually prove and establish this? He now conduct a Punnett table. They call it Punnett square. You can also use the Punnett square for monohybrid inheritance. Where we did that course, you can use a table and have your capital T, sorry here, capital T, small t, this one should not have anything, small t and small t. So it can be, this Punnett table can be used for monohybrid inheritance, but most of the time it is used for dihybrid inheritance. So what did he do? He constructed a Punnett table. So for dihybrid inheritance, you cannot use the crossing method. You use what? Your Punnett table. You call it Punnett table or Punnett square. So they used to ask that too. It is used mostly for dihybrid inheritance. So this is what we would have. So he kept all the proposed offspring that I should get. I hope we do economics? Many of you do economics. Must have done statistics. Yes. Okay, we should do it in physics. Have a plant that is yellow and round to be crossed with another plant that is what? Yellow and round. So please, I'm begging you, if you want to answer questions that requires you to construct a Punnett square, who left and is just joining? When did you leave? English people, when did you leave? I hope you've not missed a lot. So if you're answering a question that requires that you construct a Punnett table, it is always very careful. Like, it's very important that you are careful with you alternating your genes or your allele rather. So for your yellow and round, make sure it is alternating with the yellow and round here. So for the second offspring that is likely to get is what? Yellow and what? Wrinkled, right? So this one will also be what? Yellow and wrinkled. They are not with me oh. The other one that is likely to get would be what? After yellow and round, yellow and wrinkled, what is the next one? Green and round. So this one would also be what? Green and round. Then the last segregated offspring that we should have would be what? Green and wrinkled. So obviously the alternative one here would also be what? Green and wrinkled. Are we together? Can we proceed?
Yes, ma'am.
Now let's continue. Now when he did the second offspring, the F2 generation, he now got offsprings that are 16 offspring that he got. Now what is the genotype and the phenotype of those 16 offspring? That is what we would now use this Punnett table to find out. So for this first one, when you multiply your yellow round, so what would we have? We would have a plant that is what? Homozygous yellow round. Is that correct? This plant is homozygous for yellow and round. The same gene. Homozygous yellow, homozygous round. Do you agree? Are we lost? Please, if you are lost, take me back. I need response. Do you agree with that homozygous yellow, homozygous round for the first offspring? hope I've not been logged. I can't hear anything. Hey, subhanallah, time is all.
Yes, ma'am.
Okay. Then the second offspring would now be what? Capital Y, capital Y, capital R, small r. So you have your, let's take the first one here. Capital Y, capital R, capital Y, small r. So he got a plant that is what? Homozygous yellow and heterozygous what? I need your response here. Homozygous yellow and heterozygous what? Oh yeah, now talk now, so that I will not have not in this alone. We are in it together.
Ma'am, we didn't get it. I didn't get the question. Sorry.
I said the first offspring, this one. So the second offspring, this one with this one that gave rise to the second offspring here. He got what? Homozygous yellow, capital Y, capital Y, right? He got capital Y, capital Y, which is of course homozygous yellow, the same gene representing that yellow trait. And heterozygous what now? Round. This offspring will still be round, even though it is having what? A recessive trait, a recessive gene for the roundness trait. It will still be round, but that round will not be the same round here. This one is homozygous round, but this round here is what? Heterozygous round. So even though they are both homozygous yellow, this is homozygous round, this one is heterozygous round. Is that, are we together now?
Yes, ma'am.
Okay, the third offspring, we need to be fast with this, or I probably would continue from here in the next class. Let's continue. We would pick it up from here in the next class. Then the next offspring, because I have another class soon, like soon. So the next offspring would now be capital Y here now. This capital Y and this small y. Capital R and capital R. So of course we pick the Y and R together first. So capital Y, capital R, small y, capital R. So we now have an offspring that is what? Capital Y, small y. That is heterozygous yellow. It is still yellow because we have a dominant gene here, capital. So it will still be yellow. Even though the Y, small letter Y is representing green, it can't be dominant. It is recessive. So the Y is dominating over the green. So the yellow is dominating. So it is still yellow plant with what? Capital R, small r, capital R, capital R, homozygous round. Heterozygous yellow, homozygous round. Then the fourth offspring would now be capital Y, capital R, small y, small r. This one, sorry, this multiplying this. So we have both heterozygous yellow and round. Because we still have two dominant gene, capital Y and capital R, so they are still dominating over the green and wrinkled. So we still have homozygous round, heterozygous round and heterozygous yellow plants. And then it continued and continued and continued like that. Okay, should we quickly continue it, or we should just keep it and start from the beginning? We have 1 minute more and I want us to evaluate what we've discussed so far. We are going to pick it from the hybrid inheritance next week. I would be fast as much as possible with it so that we can also discuss variation. And I will send an assignment question to the platform that I would want us to attempt. But before we move on, let us summarize what we have done so far. We looked at the differences between genetics and inheritance. Genetics studies inheritance, while inheritance itself is the transmission of traits from parent to offspring, and genetics studies that transmission. We looked at some terms that are commonly used in an attempt to study genetics. Homozygous, heterozygous, allelomorph, dominant character, phenotype, genotype, dominant gene, and the likes. So please read up on all these terminologies: first filial generation, mutation, and the likes. We also looked at traits that are inherited, transmittable traits from parent to offspring. We looked at traits in plants as well. Then we looked at the father of genetics, Gregor, and what led to his experiment on genetics, why he used the Pison plant that he used, and then his monohybrid inheritance that led to the first law of inheritance, and how he conducted the experiment. And then we started the dihybrid inheritance, even though we are unable to finish it. So in the next class, please tell your friends who would complete dihybrid inheritance. We'll look at the structure of the gene, the chromosome itself. We'll look at the structure of the chromosome and the DNA that make up the chromosomes, the composition of the DNA, the thymine, the guanine, the cytosines and the likes, and how they are needed for protein formation, of how they code for any particular character. And we'll also look at the application of in genetics, and then we'll look at variation, God's willing. So do you have any question before we call it a day?